We tried to keep a little bit of mystery in the title of this talk. So I was a little bit sad when the very good question to observe came, took away a bit of the thunder, but I can tell you already now that yes, you can make shared and precision vaccine from these endogenous retrovirus targets, and this is a little bit what I've been showing today, how we do that in Evaxion. But first and foremost, let's just remind ourselves on why this is actually important. So this figure here, this is probably the most famous figure in cancer research. It was first published in 2013 by a large consortium of global researchers. And what it shows is that it shows how many mutations are there in different types of cancers. And what we see to the right, that is the type of cancers that has a lot of mutations. And then we also see that to the left, there are a number of cancer types that have very few mutations. And as Thomas was explaining, these mutations are the source of modern personalized cancer vaccines. These are the input that we need for Pioneer to find very good neoantigen sources. And as Thomas also showed, this is important for efficacy. So even though we have a great system, there might be some cancer types that is just very difficult to treat because they have few targets for these modern cancer vaccines. So, luckily, Christian, there you are. Luckily you came along and showed us that there are these endogenous retroviruses in the human genome. So these are the ones that has been inherited from our ancestors, and we all have them. And they constitute a huge part of the genome. And normally they're all silent, but as Christian also showed, these, for some reasons, start to be used in cancer cells. They're not used in our healthy cells, but they're used in cancer cells. And these are actually extremely good targets. So after realizing this, we start to look into, so what does the research literature actually tells us about this? Is there any proof that this would work in patients? And yes, there are. And if you're looking into AML patients, we see here that people have actually done a lot of great research on this. And what we see here on the left is a research group that in 2020 published some data showing that just from unvaccinated patients, they could see that the immune system of these patients actually picks up some ERV. So they find that the immune system already, without being primed by a vaccine, is already picking up on these endogenous retrovirus targets. What we see in the middle of this is basically the same story, but from a different research group, that they can actually take out blood from patients and stimulate that with these specific targets, and they see an immune response. This is quite remarkable. It tells us that we might be onto something that can be used in a vaccine. And what we see here to the right is that these are actually displayed on the surface of the cancer cells. Just as we saw in our experiments when in the mouse cancer cells that Christian was talking about, we also see that when we look into humans. So these are important data for us to make decision on going forward with these kind of targets. So the next thing is it's all about data. So one thing we have done recently is to gather a data set of more than 15,000 patients and then run them through our Pioneer model and also run it through our observed model. And when doing that, and this is just for selected cancer type, when doing that we see that, just as Christian was explaining to us, that for some cancer types we see a lot of ERV, but that is not correlated with the number of mutations. And here I have just highlighted a specific class and type of cancers, and that is blood cancers. And we see that for some of these blood cancers, they just have a lot of these endogenous viruses. And of course, these could be very interesting cancer types for us to explore further. So, now we are going into why is this so exciting? One thing we also realized was that some of these targets are shared among patients. That is very rare when we talk neoantigens, when we talk mutations. But when we talk about ERVs, this is actually a common phenomenon. And another realization is that these are just viruses. They look a bit different from what we are used to, but back in the day, when they infected one of our ancestors, they were just viruses that was then integrated into our genome. And we have already built an AI-Immunology model that are very effective in designing virus vaccines, our Raymond model. So why don't we take building blocks from the Raymond model and build that into our observed model? That was exactly what we did. So we took the building blocks that is needed to build shared and precision vaccines from the Raymond model and build that into our observed model. And thereby, we created what we call the Observe 2.0 AI-Immunology model. And by doing this, we were then able not only to do personalized ERV and neoantigen vaccines, but we were also able to make shared vaccines, where you make one or a couple of vaccines for the entire populations of cancer patients within a given type of cancers. And we took the new Observe 2.0 and just employed it to a number of promising cancer types to see, is this actually possible? And this is the results. And basically, the answer is, yes, it's possible, but I will also tell you what this figure means. It is maybe a little bit complicated. So, what we have here on the x-axis, that is number of epitopes, so number of targets that the given vaccine would target each individual patient. And what we have on the y-axis is the likelihood of the vaccine actually inducing an immune response in any given patient in that population that having this type of cancer that will then be able to target this target in the cancer cell. So what we see is that if we are just requiring the vaccine to have one target, if that is enough, that is normally not enough, then what we see, for instance, in ALL cancers and AML cancers is that we, with a very high likelihood, just with one vaccine, would be able to induce an immune response in that patients that will target at least one target. And then we have 2 targets, 3 targets, 4 targets, 5 targets, 6 targets, 7 targets. And if you look at 8 targets, we see that the likelihood is still very close to 1, meaning that it's very close to 100%. And if you then go, say, if you want 25 targets, then it goes a bit down. But what we also see is that, for instance, for lung cancer, it's very difficult to make shared vaccines. So here, the concept is much more difficult, which means that in that case, we probably need a personalized vaccines or a precision vaccine. So, another thing, we started to look into these different cancer indications. And one specifically indication we looked into was AML. So this is a blood cancer that unfortunately hits children, but also adults. And when we start looking into the data that we had one data set for children, and we had another data set for adults. And we wanted to see, so do they have the same targets, or do they have different targets? And when we start looking into that, the first thing we did was to look, do they have the same number of ERF expressed? And what we see is actually that the children, if you are a child below 20 years old, is being diagnosed with AML, you have much lower number of ERF expressed than if you are an adult above 20 years old. So these could be two different diseases, actually. Also, what we investigated was that whether this expression of ERF correlated with disease states, so how severe the disease are, and that was not the case. It simply only correlated with age. So there are some interesting aspects in that we wanted to explore even further. So we looked into what ERF was actually expressed in adults and in children. And what we saw that it was basically the same ERF, but adults just expressed more. So they have a core that is expressed, and then if you are an adult and diagnosed with AML, you simply just have more ERF expressed. So we took our Observe 2.0 model and said, okay, but could we use this information to make even better vaccines? And then what we did was that, why don't we do one vaccine for adults based on the ERF expression in adults, and another one for children? And what we see that if we do that for the adult population, it doesn't make a lot different, because they are already having all the ERF expressed that we were targeting with the fully shared vaccines. But if you look into children, and just look in the case where we want 25 targets, here there are a huge difference depending on whether you make shared vaccines for all AML patients or a precision vaccine based on which ERF actually expressed in these different patient populations. And this is of course something that we are going to explore even further in other indications. The split doesn't have to be in children and adults. It could be based on co-expression of ERF. It could be based on HLA types, so what kind of different immune system do we have? So right now we are exploring how we can use this platform even further to make new pipeline products. So my job currently is to explore how can we make this as fast as possible into the preclinical stage, and of course also later on into humans. And with that, I hope that I convinced you that, again, ERF is a promising target, especially for these cancer types that are difficult to treat with our current modern vaccines. Also, we have developed this Observe 2.0 model that have building blocks from Raven, which allows us to do these shared or precision vaccines. And of course, these are great new avenues. I think we had a question before on the manufacturing cost, on personalized medicine, and of course, if you have a shared approach, the approach to market is different from when you have a personalized approach. So with that, I hope that I've just sparked your interest a little bit in this, and just maybe open up the window into what is coming next in the field of cancer vaccines. So with that, thank you.